Please join me in welcoming to Distinctive Voices, Dr. Rita Colwell.
It's a great pleasure to have an opportunity to talk to you tonight about my favorite subject, the interaction of the oceans, climate, and human health. And I will discuss particularly the theme of my talk is the interaction of the environment and human health. And I take the words of Hippocrates from the 4th century BC, who said, "Whoever wishes to pursue the science of medicine must first investigate the seasons of the year and what occurs in them." So that's the general theme of my talk. And I will end up discussing a very, very timely and tragic situation, namely cholera in Haiti, and give you some of my ideas based on 40 years of doing research on cholera and the environment. Water quality is really critical, and it is demonstrated, I think, most dramatically in children's survival. Children under the age of 5, if you look at countries like Cambodia and Ethiopia, where less than 20% of the population has access to safe drinking water, the deaths of children are like in the range of 200 deaths per 1,000 for children under the age of 5. So there's this really direct correlation to safe water and proper sanitation. Cholera is a global disease. I'll be speaking about it quite at length tonight. It's an acute water-related diarrheal disease. And we talk about pandemics. We're presently, according to epidemiologists and the way we sort of look at the waves of cholera outbreaks, in the 7th pandemic. It began in the 60s. It now has covered more than 50 countries, and about 7 million people have been afflicted. Bengal, the Bay of Bengal, and Bangladesh, India, are considered the home of cholera. But I would put that in quotation marks because a very nice book was published a couple of years ago called The Biography of Cholera. And it points out that we had cholera in Europe, in the United States up until 1900. It's just that it still has not been eradicated from the developing countries, particularly Bangladesh and India. But cholera exists in the natural environment. That's been a discovery that my laboratory made about 30 years ago, but it's been very hard to penetrate the psyche of the medical establishment. It's a bacterium that I could isolate from this bay right outside this room with appropriate microbiological media and techniques. It's a bacterium that is associated with plankton. It's a gut flora of the plankton, just as we have E. coli and specific bacteria in our gut that helps us digest food. And as we're finding in more and more, it affects us in many, many ways that we hadn't expected, even neurologically. So for the zooplankton, particularly, the Vibrio cholera, the bacterium, is associated with it, part of its natural flora. And therefore, it's unlikely that we can eradicate cholera simply because it's part of the environment. Almost all of the strains of cholera digest chitin. That's the structure of crabs, shellfish, crustaceans. And about 20% are bioluminescent. So, and these bacteria also carry out functions in the environment in carbon and nitrogen cycling. So they're part of the natural environment, except that when ingested by humans in contaminated water or food, then disease ensues. Now, the bacterium that we culture, shown here, it's a little rod-shaped bacterium with a little tail, a flagellum. And when we grow it on specific media, it's a bright yellow colony because it ferments sucrose and therefore produces acidity, which then causes the color changes. Now, this is a cartoon from the New York Times about 1850. And It shows science. If you read the belt of the soldier, it says science asleep while the specter of cholera comes into the United States. We've always considered cholera as coming from somewhere else rather than actually occurring seasonally, as I hope I'll show you tonight. The bacterium also undergoes a very interesting phenomenon of dormancy. It's a bacterium that does not form spores or resting stages, and it was not considered to have a resting stage. But we discovered about 20 years ago that the bacterium actually, when suspended, these are actively growing bacteria, but when suspended in a saline solution, put in the refrigerator, after a month they begin to shorten. After 2 months they round up, and then at 6 months they're really essentially spore-shaped, and they can resist changes in temperature and so forth. It's been a phenomenon that's been very hard to get established in the literature initially, but now there are more than 1,000 publications on this phenomenon in the bacteria that don't form spores. We refer to them as the gram-negative bacteria that don't form spores, but they do have this resistant stage. Now, we were able to discover the fact that these bacteria could be found in the environment when they couldn't be grown in this dormant stage by using an antibody very specific to the surface of the bacterium and then linked to a fluorescent molecule, sodium isothiocyanate. And then you look at it, the smear under the microscope, an epifluorescent microscope that allows then for the dye to glow essentially, and so you can see the bacteria very clearly when you cannot grow them. Now, we just discovered this in 1980, and the Centers for Disease Control, CDC, referred to it as Colwell's ghosts. However, now even the CDC has published a paper on the viable but non-cultural stage of gram-negative bacterium. We discovered the relationship with plankton about 1975, and that has been very instructive. We find that this is a copepod that's found in Chesapeake Bay, where we did our early studies. And this is the egg sac of the gravid female. And then the surface is coated with bacteria. When you look under the scanning electron microscope, you see lots of bacteria. These bacteria, Vibrios, also produce a very powerful proteolytic enzyme, probably associated with the zooplankton because these copepods cast their eggs into the water column in the spring. And very likely, the bacteria play a role in producing this proteolytic enzyme that causes the splitting of the egg sac when it's ripe, so to speak, and then the eggs can be cast into the water. This kind of an association has been described by other scientists who are expert with zooplankton. Now, we did studies in Bangladesh at the time, 20 years ago, when it was still refuted that these bacteria, the cholera bacteria, could exist in the environment because they couldn't be cultured. They were in a dormant stage. And we were able to show with this antibody dye reaction, and this is a copepod in a pond in Bangladesh during a cholera epidemic when the families are drinking the water directly from the ponds that have these zooplankton covered with the cholera bacterium. So in this brief summary of kind of the life history of the bacterium, we can see that particularly the copepod is the source of the bacteria. Other organisms, oysters, crabs, fish, even chironomid flies have been shown to carry the bacteria, but the natural association or the commensal relationship is really predominantly with the zooplankton. Now, cholera is a disease that is very rapid onset. It's a diarrheal disease with severe vomiting and watery diarrhea. In fact, this rather crude technique of measuring the fluid lost is still used. in Bangladesh. It's crude, but it's effective. When I first did the work in Bangladesh, it was in 1974-75, and at that time the hospital was simply a cement slab and a canvas roof, and then during monsoon you just roll down the walls, canvas walls of the hospital, and the cholera cots were as you see here, and a victim being brought in during monsoon, the woman's husband is carrying her into the hospital. But the hospital, fortunately, is now much improved. This is the amount of fluid required for a severe near-death case of cholera. You can lose 18 liters in a day, and in fact, the diagnosis is what's called dry skin. You can actually pull with your fingers the skin of the abdomen, and that is an indication of a severe desiccation. And so to replenish, it's usually done now by a very dramatic discovery made in India of what's called oral rehydration. It's simply sugar, table salt, bicarbonate of soda, mixed in appropriate quantities in a liter of water, and then sipped. Now, you can buy this now if you have small children, and they have severe diarrhea. It's called Pedialyte, and it's available in drugstores. So that is what was originally developed to treat cholera, and developed by an Indian scientist, medical doctor, about 30 years ago. So we also worked out a relationship of these bacteria in the environment to understand the seasons, the effects of climate on cholera epidemics. Because in Bangladesh, the epidemics are like clockwork. May, June, July, you have a peak in the number of cases. Then the monsoon hits and there's a hiatus. And then September, October, November, there's an even bigger peak. Year after year after year. And so obviously, to those of us who are environmental scientists, it shows that there must be a relationship between climate and this massive epidemic that occurs in these countries every year. So in studies that we did, first in the Chesapeake Bay, and you may say, why Chesapeake Bay? We don't have cholera. Well, it turns out there is a seasonality. You can detect it in the bay, spring and fall, using molecular techniques. So it's there, but we don't drink the water straight from Chesapeake Bay without filtration, chlorination, safe distribution. So we don't have cholera. But nevertheless, the bacterium is still in the environment. And so with the studies over a 30-year period, we found that sunlight, temperature, nutrients, salinity, and rainfall all play an important role. Because what happens is, and this particular community understands the environment so much better. You know a lot more from your lectures that you've probably attended in the past, that these factors lead to increases in the phytoplankton, which use sunlight as an energy source, and then the zooplankton feed on the phytoplankton, and then the zooplankton carry the bacteria, which when the zooplankton population just crashes, then there's larger numbers of the cholera vibrio in the environment. And it's an infectious dose is about 1 million of the bacteria per teaspoon. And this was developed in volunteer studies about 50 years ago, which you couldn't do now. The Jessup prison in Maryland was the source of the volunteers, and it was the University of Maryland Medical School, not I, it was before my time, who determined that this is a dose-dependent disease. 1 million cells per milliliter, and you will get full-blown cholera. And if you have only a few hundred cells, then you may get a mild diarrhea, 1 or 2 stool diarrhea, and that will be about it. So there is a relationship to numbers, and that's important. We were able to show that just from all of the data that we gathered on temperature and salinity and nutrients and all the factors from the environment, just measuring environmental factors, we were able to build a model. And this model shows you that the red line is what we predict based on all those factors taken into account. And this is for the Bay of Bengal, and this is for Bangladesh, Matlab, an area in Bangladesh where we've done our studies over the last 40 years. And the columns are the actual cases. So you can see that the model fits reasonably well, except for this one year when the prediction was low, but the numbers were higher based on an unusual monsoon that year. But otherwise, the model fits reasonably well. Now, summarizing then the environment, the bacteria, the fact that they're associated with plankton, and the plankton are fed on by the shellfish, in a population that does not have access to safe water, there is person-to-person transmission, no question. But really, the environment is the source of the bacteria in the first place. Now, what about macro-scale processes? What about global climate and cholera epidemics? The outbreaks, as I've already pointed out, the work that we have done has been associated with temperature, salinity, etc. Now, this gives you a view of cholera globally. The darker the color, the darker the red, the more intense the epidemics. You can see that in Peru, in the 1990s, they had a severe epidemic. Mexico constantly has cases of cholera, as do some of the countries in Latin America. But we've pretty much considered this particular area of the world, India, and especially Bangladesh, and the effects of the Himalayas and the Gangetic Delta that feeds into the Bay of Bengal. This region, and particularly Matlab, which is the area just near Dhaka, the capital city of Bangladesh, where we've done our work over the last 40 years. And you can see that here the red spots are the hot spots of cholera in this little region, and the blue were the ponds around which the families have built their homes. Now, the seasons of the year, we have found by satellite studies, measuring sea surface temperature, and these are NASA compiled over several years that you're seeing, the changes in sea surface temperature, the changes in sea surface height, because tide plays a role as well, bringing salinity into the river system. And the effect is very dramatic and very important on the plankton populations. Now, we can measure chlorophyll by satellite. Those sensors will allow us to measure chlorophyll. So you can see during the year, each year, the spring, fall, accumulations of plankton. And that has been, to our thought, could be correlated with these seasonal epidemics that occurred in Bangladesh. And so the studies that we did, particularly in the Bay of Bengal, in this particular region here, we found that indeed, and this was the very first study that we did back in 1987 and 88, we were able to correlate the chlorophyll measurements, the sea surface temperature, shown here, sea surface temperature in blue, and the actual cholera cases in Matlab, Bangladesh. Now, consider that you're measuring the sea surface temperature miles out, and you're essentially counting feet, patients being brought into the hospital. And that correlation was very dramatic. We published those data in the late 80s in the Proceedings of the National Academy of Science. And at that time, it was considered that we were sort of stretching things a bit in trying to relate satellite imagery with being able to predict outbreaks of disease. But I can tell you since then, there have been hundreds of papers, and now there are about 10 laboratories around the world who are studying the relationship of satellite measurements of environmental factors and malaria, as well as cholera and other human diseases. In fact, our more recent work has been even more dramatic in that we've been able to show Calcutta, or is now called Kolkata, Madla, Bangladesh, Kolkata, India, a relationship between our more perfected model, the predicted cholera cases and the actual observed cholera cases. And you can see the relationship as our prediction has improved. And again, another Proceedings of the National Academy of Sciences publication in 2008. Now, these results, by using satellite measurement of chlorophyll and then ground measurements of rainfall for Calcutta and MATLAB, we've been able to show that with a milligram per cubic meter increase in chlorophyll measured by satellite, you get a 33% increase in the number of cholera cases. By a millimeter per day increase in rainfall, you get a 7% increase in the number of cholera cases. We've extended these studies, and we're just now publishing a paper on similar kinds of analyses for Senegal in Africa. And one of my former postdocs, Guillaume Constantine de Magny, who is now back in Montpellier, has published a paper on similar kinds of predictive results for Ghana. So we now know that in Africa, Asia, and even in Chesapeake Bay, we can predict by using satellite measurement of environmental factors. In Bangladesh, the milligram per cubic meter of chlorophyll measured by satellite is a 32% increase in the number of cholera cases. So these relationships are getting finer and finer and more highly predictive, which is rather exciting, actually. Our current research is to take into account in our models flooding as well as exposure and immunity in the population, as well as these other extrinsic factors, the intrinsic human factors, we're building into the predictive models that we're working on. Now, I'd like to talk a little bit about genomics, because it's been enormously instructive for understanding disease outbreaks. Back in 2000, it's 12 years now, gosh, time does fly, my student, John Heidelberg, was on a postdoc at the Institute for Genomic Research in Rockville, Maryland. And we sequenced, along with John Mekalanos at Harvard, my team at Maryland, and Tiger, for the first time, the chromosomes of Vibrio cholera, the causative agent of cholera. And we confirmed that it has 2 chromosomes, which was interesting to microbiologists, because until then, it was assumed all bacteria just had 1 chromosome. Now we know from many other sequencing that's been done that Vibrios and some of their relatives, like the photobacteria and the aeromonads, also have 2 chromosomes. But this was very interesting for us at the time, a large chromosome and a small chromosome. And with more recent work, we've actually been able to follow the evolution of the genes in the bacterial chromosomes that have manifested by changes in serotype. And serology has been used as an epidemiological tool for well over 100 years. And one of our surprises was that we found that the genes that code for the serotype are laterally transferred. So the epidemiologists really have been tracking the genes coding for the serotype rather than the bacterial cell in toto. And there's a great deal of lateral exchange of genes. They're very promiscuous, these little bacteria. And so almost 70% of the genes can be laterally transferred, and maybe only about 30% of the genes are what are called core genes that are sort of stable. And so shown here is the fact that the genes that code for the antigen and the oligosaccharide that comprise the O1 serotype for Vibrio cholerae are genes that can be laterally transferred and also are subject, of course, to mutation. And we've been able to show in the last couple of years that, in fact, there are changes that occur with what are called the pandemic strains of Vibrio cholerae that undergo genetic drift and genetic shift. Terms that you probably have heard about influenza, how it can drift and you have to get another vaccine year after year. And it's because the genes are moving around a bit. And we find that that also occurs in the cholera bacterium, which may explain why it's been so difficult to get a, shall we say, a vaccine that's highly effective against cholera. Now, I'm going to go into probably, maybe it's a little esoteric, but it's very interesting. Proof that the environment is the reservoir of cholera. And you may think that I'm sort of pushing this to the extreme, but you have to understand that there's still the naysayers who will demand that it can only be transmitted person to person. But the environment is really the reservoir. This is a paper we published just recently. But our biggest surprise was my being able to do a dive on Alvin, your very own Alvin that's just down the road. And actually, I just visited it today to see the new Alvin that's going to be working very soon. But I did this dive when I was director of the National Science Foundation. It's one of the perks. You get to do really fun things. So I did this dive on Alvin, and of course my friends asked me, was I nervous? I said, no way. Woods Hole did not want the headline of the New York Times saying, NSF director lost at sea in Alvin. So I felt comfortable. Now, when I was on Alvin, I could see that there were little things sort of swimming around. And it occurred to me, are there deep sea copepods? I didn't know that. I should have, if I'd been a good marine biologist. And the answer from my colleague, the scientist who was always also with me on the dive on Alvin, said, yes, there are. So we took samples, and we were able to bring them back on deck. Actually, the particular dive was to study methanogenic bacteria, which grow without oxygen. They're anaerobic. But study had been done in my lab some time earlier showing that Vibrio cholera does grow with no air. It does grow anaerobically. In any case, we were able to isolate this bacterium, which we numbered EX25. And this was from one of the chimneys in the deep sea. And we were able to show that this bacterium grew nicely on that media, produced those yellow colonies that I showed you earlier. And then when we extracted the DNA, and then with the sequence lined it up with the other bacteria, we found that it fit right into the human pathogenic Vibrios. So here we have bacteria from the very deep sea, 200 miles off the Oregon coast and a few miles down, that have the genes that confer pathogenicity. So our conclusion is that these genes, which are shared, this deep sea bacterium, with these human pathogens, Vibrio perihemolyticus and alginolyticus, and also some genes with the cholera bacterium, it occurred to us that what we're dealing with, that we call pathogenicity genes because they make us sick, are really ecologically powerful genes for the bacteria. That is, they have a role in making the bacteria ecologically fit and competitive. Now, another study, which we just published last year, was done in Iceland. One of my students, former students, Dr. Brad Haley, who's now with the USDA, did a Fulbright in Iceland. Now, cholera has never been reported in Iceland. It's a reportable disease. In fact, they have wonderful records going back to the actual founding of the country, of the various genealogical records and the health records. And they do record pandemics that have occurred, and they've had a plague outbreak and a flu outbreak. So It's not that they don't have good records, as is the case in Haiti that I'll discuss in a moment. It's just that it doesn't occur in Iceland. Now, the student took samples at these geothermally heated sites. He also took samples at sites where there wasn't any geothermal heating. There's Brad busy taking samples and trying not to fall into this very hot geothermal discharge, and then compared to an area where there was no geothermal heating. And he was able to isolate Vibrio cholera from Iceland just from the geothermally heated sites. The plus means Vibrio cholera was detected. So here's a cholera bacterium in Iceland. They never have cholera. And so what are some of the reasons why not? Drinking water originates as rainwater in Iceland. It's collected, it filters through basaltic rock, and then it's heated. So it's essentially boiled and then distributed to households. Icelanders don't eat shellfish. They have mussels there. They don't have oysters, but they have mussels. Mussels are used as fish bait. They don't eat it. Just a culture, I guess. And there's not very much interaction with the water. They don't swim in the cold beaches. They do have swimming pools, so they swim, but they don't swim in the coastal waters. So essentially, people in Iceland don't interact with the marine estuarine environment directly, nor do they consume food from that environment. Why do those bacteria have all these pathogenicity genes? Well, we think they have a role in the natural ecology of the cholera bacterium. And the evolutionary roles are not for human infection. It's just that when we ingest it, if I may put it colloquially, the bacteria behave as if we're just one great big copepod. And they're trying to help us osmoregulate, which means our sodium-potassium imbalance is completely upset. We end up with huge flooding diarrhea and vomiting, and we become dehydrated and we die. So it's a matter of an inappropriate host, and yet the pathogenicity factors, in fact, one of the set of genes confers pressure tolerance, which explains why the deep sea habitat is a fit one for the cholera bacteria, cholera-like bacteria that we hauled up on the Alban dive. Now, the genomic diversity of cholera is very important, and it allows us to understand the Haitian cholera epidemic. That's been a huge tragedy. The outbreak initially, ostensibly, was near this region. Actually, I can show you the outbreak site, presumed to be about here. And very rapidly, the epidemic just really broke out throughout the whole country. Within a matter of 3 or 4 weeks, cholera had completely covered the country of Haiti. Now, we were able to obtain in November, let's just go through what happened in Haiti. First, in January 2010, there was a massive earthquake. I think it was something like 7.8. Very, very powerful earthquake. It's a country that the river system and its geology is such that the rivers are fairly alkaline, at least from the measurements that we've taken. And in the summer, it was a very hot summer. The temperatures were up to, in the water, to 32, 35 C. I've been challenged that it wasn't hot, and a French group had taken about 10 years of incomplete data and said, nah, it wasn't hot. But we took 50 years of data downloaded from the NOAA database and from NASA, and showed statistically that it was one of the hottest summers on record. And then, as if those poor citizens of that country didn't have enough, the third horse of the apocalypse arrived in the form of a hurricane just offshore that caused tremendous flooding. And then the fourth horseman of the apocalypse arrived in the form of a massive cholera epidemic that started probably around the 12th, but some say the 21st of October. We were able to obtain samples of clinical samples from 81 patients through the good auspices of a physician who was working with a nonprofit religious group who had gone in to help with the earthquake disruption. And so that doctor and his nurse provided us. We shipped in because they have no real clinical lab, and they don't collect data. They don't do the usual tests. So we flew in the media, the test tubes, and all of the gear, and then the physician then from all of these places in Haiti, we were able to get samples and then extract the DNA from the bacteria that we isolated from those clinical samples. So here are the patients from the different towns, the arrondissements, from Atibonite and from the northwest and the west part of the country. So we were able to get full samples and analyze them very extensively. And we discovered something that's been contentious. We published it 3 or 4 weeks ago in the Proceedings of the National Academy of Science, because we found that 48% were the typical classical O1 serotype of cholera, the pandemic serotype. But we found that 21% were not that serotype. Furthermore, 7% had a mixture of the pandemic serotype and the non-pandemic serotypes, and then 24% didn't even have the cholera bacteria at all. So we have published the data, and of course we're getting a great deal of pushback because, as you may have read, initially when the outbreak began, 2 or 3 strains were isolated by CDC, and they did they didn't do a full sequencing of the DNA, but they did some of the comparisons, and they said it was similar. I give them credit. They said it was similar to Asian strains of cholera. And then another team did some sequencing and said it is identical. Well, they didn't say identical. They said it is very closely related to the Nepal cholera outbreak strain. And immediately there was a rush to judgment that the Nepal troops had brought in cholera to Haiti. And so, as you may remember from reading the newspapers, the natives, the Haitians, attacked the Nepal troops. There were fights and riots, and some deaths occurred, which is very unfortunate. There were many other troops besides the Nepalese peacekeepers. There were some Bangladesh and some other countries. I don't remember all of them now. But only 15,000 of the Nepal troops at any one time were in Haiti, and none of them had been tested positive for cholera. So our conclusion was that it may have been introduced, but it is much more complicated. And this gets a little sticky because there's now a lawsuit by a nonprofit group in Haiti. That group in Boston is assisting to sue the United Nations for reparations for having brought in the cholera epidemic. So my view is, let's step back and get more data and understand this a little more completely. Our results show that the Haitian cholera strains clustered with other pandemic strains, but they formed their own independent cluster. And strains from Zimbabwe, Bangladesh, Thailand, Zambia, Mexico, fell into that same kind of general relationship. In fact, if we did a principal component analysis and showed that the Haitian strains formed a separate cluster, and some of these other strains from other countries that had been involved in outbreaks in those countries were similar, and perhaps as similar as the Nepal strains. The Haitian cloud is shown in this three-dimensional plot, shows the divergence of the DNA from of the various strains that have been sequenced. And if you do the phylogenetic analysis, we find that the Haitian-specific strains form this cluster, and this is a Bangladesh strain. This is another Bangladesh strain. This is the Zimbabwe Africa strain isolated in a 2009 epidemic. This was a Bangladesh 2010 epidemic of cholera, and this was Bangladesh 2002. This is a Thailand outbreak of cholera in 2010. And then, ironically, this was the original strain that we had sequenced that the CDC said was similar to the outbreak, and hence an Asian strain. So it's a bit more complicated, but what I'd like you to have as a takeaway is that with genomics, we can now do attribution in a very, very elegant way in outbreaks of disease, and that this tool is a really dramatically useful tool. And our conclusion is that there are at least two distinct populations of cholera vibrios that have been contributing to the outbreak. And it may well be that the clonal O1 strain precipitated it, but the evidence still yet has to be gathered. And these other comments are that these populations in Bangladesh have a genomic backbone that's similar to the toxigenic strains that are circulating in the Western Hemisphere. And these are more evidence of the microbiological and genomic type that shows that the genomic analysis allows us to do a very sophisticated determination. Now let me turn to another way of looking at this cholera story. I've given you stories of results of our understanding the environment, the bacterium existing as a natural component of the environment, the ability to use environmental factors and satellites to measure how we can predict outbreaks, and then the elegant genomics to show how we can trace the outbreaks. What good does that do the person in Bangladesh who has to drink that water and doesn't have safe water? And it occurred to us that if, in fact, we were right, that the plankton carried these bacteria, we figured that if we could develop a very simple filtration, we could reduce cholera. And so when you're working in these villages in Bangladesh, these are really huts, and the families, the mud floors and so forth. And they take water, the women go down 6 a.m. in the morning, and they collect in these calashes the water for the household for the day. So we tested Chinese poplin cloth, we tested men's t-shirt material, and we tested fancy sari cloth, and we found that the very best filter was used sari cloth that had been discarded, no longer worn as a dress by the women, but were used as dust rags. And if you folded them 4 to 8 times, you got a really nice 20-micron mesh filter. And the plankton are 200 micrometer, I should say. My husband's a physicist, and it's not micro, and he says micrometer. Anyway, the bacteria are 10 micrometers, but they attach to the plankton, and they attach to particulates. And so what we did then was to test it in the laboratory, and we found that indeed we could remove almost 100% in almost every case of the zooplankton. Not so effective with phytoplankton, effective but not so effective. But then the phytoplankton are not the major carriers. We were able to carry out a study funded by the National Institutes of Health, though initially, I have to say, it's always fun how you do these science projects. We submitted the proposal. First of all, the National Institutes of Allergy and Infectious Diseases didn't think it was sophisticated enough, so they lateraled it over to the Nursing Institute. That's okay. Thank you, Nursing Institute. You were wonderful to support us. But the first review, one of the reviewers said, this isn't going to work. Men will not drink water filtered through sari cloth because it's, tainted by female whatever. So the Thrasher Foundation gave us $100,000 to go in and do a trial. And amongst other observations we made was, first of all, it was effective, but men had already been using just a single sari cloth to filter the flies from their beer. So my advice to young students is, when you get your reviews back and your proposals rejected, revise it and resubmit it. We did a very careful study. The layout was done by Dr. Estelle Russick-Cohen, who she was at Maryland for 20 years, taught all the stat courses, and now runs the Statistics Bureau at the Food and Drug Administration. And Estelle laid this out very carefully with the sari filters, filter group of households, and then the control groups that we didn't provide any education or any training. And then because in Africa there had been the use of a nylon filter to remove the copepods that are in Africa, the carrier of dracunculiasis, and it had been effective in removing and reducing the incidence of that disease. We used the nylon, but the trouble is that it costs about $10 a yard, and that's a month's salary. So, but we used it as a comparison. And the way we worked it was that we educated the women to serve as extension agents. We paid them a modest salary, and they went out every week to the villages to reinforce that you fold the sari cloth, put it on the mouth of your calash, pour water in, then take the sari cloth out and rinse it and air dry it because the sunlight, UV, disinfects. And so this process was a 3-year study, and it was effectively carried out by the women. And it wasn't difficult to explain to them that this is what it looked like after filtration, and this was before, when you could see things swimming around. And we explained to them that wasn't good for their children. So it was a very clear demonstration. And we were able to reduce cholera by 50%. And that was statistically significant. And we tracked every case of cholera that occurred in the families where they did use sari filtration, and found that it occurred because they had gone to visit families where they didn't filter, or they didn't always necessarily filter that one day or whatever. So we feel very confident that we were able to take these 30 years of research, of highly sophisticated research, and evidence-based recommendation for a mechanism to assist those who have no other way of having safe water. And we published this again in PNAS, and we went back 5 years later to see whether this was sustainable. Would they continue to filter 5 years later? And we found that indeed the sari villages, 75% were still using sari cloth. Some were using some other kind of filter. The nylon villages were not as effective. Only half of them continued, and they used another quarter of them used another filter. But this was the surprise. The control filters, the control villagers had learned about this, and so they were filtering, which made it very difficult for us to determine effectiveness because the controls were filtering. But one of the discoveries we did make that we could statistically determine, and we published, was that if you were a family that didn't filter, but you were surrounded by families that filtered, you were protected. This is a phenomenon that's called the herd, H-E-R-D, herd effect, and it's well known in vaccinations. If you vaccinate families, and the ones that are not vaccinated may be in the midst of families that are vaccinated, then the likelihood of disease is much reduced. So we saw this herd effect with filtration. And again, we published that a couple of years ago. One last point I want to make is probably, I don't want to overburden you, but I do want to give you something that we haven't yet published, but it is very, very interesting. And that is predicting disease using genomics. You have a patient, and we have controls, where in India, in Calcutta, in the cholera hospital, we have an arrangement where we receive from 50 patients and from about a dozen controls, that is, cohorts with no manifestation of disease. These are patients who come in to be treated for cholera. The laboratory there does 26 tests for viruses, parasites, and bacterial pathogens. They extract the DNA from that clinical sample, the stool specimen, and send it to us blinded. And these are the pathogens: cholera, Ehrlichiosis, Campylobacter, Shigella, Salmonella, viruses, Cryptosporidium, Giardia, and so forth. And one of the things that we have found is, and this is a little complicated, but let me just explain it. What we have found is that in the healthy controls, we can pick up small amounts of pathogens, probably carriers. And in the patients, they don't just have Vibrio cholera. They have 3 to 10 other pathogens. So we're beginning to think that the notion that has served us well for 100 years of finding the single pathogen that causes the disease, that we have to rethink it. And it may well be that what lands someone in the hospital may be a community of infectious agents rather than a single infectious agent. And so the results that we've got show that patients, let's just say, to try to show it a little more simply, that in a healthy patient, they'll have a whole lot of these kinds of bacteria, and you can't read the names, doesn't matter. These are benign kinds of bacteria. But the sick person doesn't have as many of the good bacteria, and it has a whole lot more of the bad ones. So when you compare these, you can see a difference between the kind of combination of bacteria that comprise the person coming in that's ill. And we can show, I'll just summarize quickly, a few things. We now know that the microbial flora, the gut bacteria, not a big surprise, of healthy humans in India is very different from us Western Europeans. We also know that the Indian population may tolerate a low number of pathogens, but I have a feeling if we'll look in the United States as people admitted to hospital with infectious disease, we may find that it's a community rather than a single strain. And we can pick up multiple pathogens from the disease patients. And so the profound watery diarrhea we now know is a mixture of other pathogens. In fact, if Giardia is present, it's been shown that the cholera bacteria produce more toxin. So that interaction is powerful. I'll conclude by quoting John Muir, one of our very good heroes, who said, when one tugs at a single thing in nature, he, and I would add, and she, finds it hitched to the rest of the universe. And there are a lot of students from the university, from Bangladesh, from other countries, from NASA, from the Los Alamos, and just a number of places to whom I extend my thanks and express my appreciation. Thank you very much.